All right, welcome back to the Jepson workshop on fungal communities. And in section 5 here, we're going to talk about the dimensions of ectomycorrhizal fungal communities. What I'm going to talk about in this section are just the basics of the ectomycorrhizal symbiosis, and particularly the plants and the fungi involved, where they are and what the interaction looks like. Okay, so there are two main kinds of mycorrhizae, and I think early on somebody was asking a question about don't all plants have mycorrhizae? And the answer is yes, they do. That with rare exception, plant roots are associated with fungi. Most plants are associated with what we call arbuscular mycorrhizal fungi, or what used to be called endomycorrhizal fungi. And so those would be, grasses and ferns and herbs and actually a lot of the trees, most of the trees. The fungi involved are not so visible. You wouldn't run into them in the forest, for example. And so I'm not going to talk about those. The ones I'm going to talk about are the ectomycorrhizal fungi. So this is a small subset. Completely unrelated fungi and a completely different but similar interaction with the roots. So this is a diagram of the ectomycorrhizal symbiosis that comes out of Anders Dahlberg's dissertation. I love this diagram because I think it captures so much. And what it shows is that you have a basic commodity exchange here, that the tree is giving sugars to the fungus, and in return it's getting nutrients, and that interaction is happening at this little colonized root tip. So we'll come back to this diagram at multiple points, but let's look at what it actually looks like in reality. So here is a small tree, pine tree growing in the greenhouse it was, and here's the genus Leucaria fruiting right next to it, which is mycorrhizal on its roots. And if we looked at those roots, you'd see a little fluff of fungal hyphae around them, and the root itself is slightly swollen. And if we did a cross section through this, you'd see that there's a wrapper of fungus around this called the mantle. And so that's what an ectomycorrhizal interaction looks like. There can be very extensive mycelium, and that's what you're looking at in this case. This is the same picture that I think Elsa showed earlier with a little pine tree, and all of this mycelium is from an ectomycorrhizal fungus called Suillus that we'll hear about more in the next section. And then the fine interaction here is where these little tiny roots are swollen, covered with a fungus wrapper called a mantle, and then the hyphae would extend out from there to make this big mycelial mat. This would be a blow up of the colonized root tips. And then what it would look like in cross section in a diagram is that these are all root cells, but this outer layer is all fungal. It's a wrapper, again, called the mantle, that goes around these roots. And then the the fungus goes between the cells, but doesn't penetrate them. So the other type of mycorrhizae that is more common actually penetrates cells and doesn't have this wrapper. These you can actually see for yourself if you dig up roots under a pine or something and look at them with a hand lens. You can see this sort of thing going on. They're macroscopic in that sense, which is not true of the other mycorrhizal type. Okay, so we just looked at this, but we didn't really, because the one thing about this diagram is it's a simplification, right? It's showing one of these. But what really goes on is when you look under a tree, you find many of them. A single tree might have several dozen mycorrhizal species associated with it. And over a very small section of the root system, you'd see multiple species. So here are the different colors and shapes and so on are telling you that it's not all the same fungus involved. And so if we go to the next level, so at a tree level, you've got, several dozen species involved. We'll go to the next level here. Now we're looking at a diagram of the ectomycorrhizal species found in a fir forest in the Sierras. And this would be about a 4-hectare area. And what you're looking at is what I call a ranked abundance curve, where species are arrayed on the bottom here. And then what you're looking at is their biomass here. And then we put them in order of the most abundant to the least abundant. So you can see there's quite a few fungi here. And we haven't given the names, but you can see there's lots of fungi here. But it took the first 29 fungi to account for half the biomass. And this is just a piece of this broader ranked abundance curve that goes out to over 90 species. And it's not a saturated curve. That is, we could go back to this forest, sample more, and find new species that we didn't see the first time we sampled. So below ground, it's like the tropics. It's hyper diverse. That above ground there weren't a lot of different plants, but below ground there are hundreds of mycorrhizal species in this single forest. If we look at global distribution of mycorrhizae, of ectomycorrhizae, that's the color bar here shows what it looks like. That basically they're most abundant in the temperate zones. And that's true both in the north, especially the north temperate zone, but also in the southern temperate zone. We can get them in the tropics too, but they're just less abundant there. Most tropical trees are not ectomycorrhizal, but there are a few that are, and I'll come back to those. And this paper that diagram came out of, I really like this quote. They say, we estimate that ectomycorrhizal trees, which represent only 2% of all plant species, constitute approximately 60% of tree stems on Earth. How can that be? Well, it's because they've colonized the dominant tree species in the temperate zones. And so they really own the temperate zones. And there, the diversity of trees is not huge, but the number of stems is. Okay, so who are these trees? And I should say, for the most part, these hosts colonized by ectomycorrhizal fungi, it's a requirement to grow. That if a tree is ectomycorrhizal, if it does not have these fungi, it will not grow. It may be able to hang out for a little while, but it's not going to get any bigger. So the poster child for ectomycorrhizae are the Pinaceae. The whole family is ectomycorrhizal. And so here's a lodgepole pine forest from the Sierras, and that would be densely ectomycorrhizal. Any kind of pinaceous tree is going to be ectomycorrhizal. Many hardwoods are ectomycorrhizal, the whole Phagales. So oaks, chestnuts, birch, alders, southern beech, all these things are ectomycorrhizal. Eucalyptus, and many, but not all members of the Myrtaceae and the Casuarinaceae are also ectomycorrhizal. And in the tropics, the Dipterocarps, a tropical family mostly in Southeast Asia, but a few elsewhere, and many tropical legumes, things like Disambi, are ectomycorrhizal. And then there are a few things that are not big trees that are ectomycorrhizal. Small shrubs such as cistus, arbutus, arctostaphylos, and willow are ectomycorrhizal. And you tend to find these, well, I'll come back to where you find all of these in a moment. Okay, so let's go to the fungal side of this. It turns out that over 20,000 species of fungi are ectomycorrhizal. And in nature, these fungi also require a host tree to grow. That is, you won't find them free living. They need this interaction, just like the plant needed the interaction. They can form big, large, obvious fruit bodies, and a lot of our most choice edibles from forest settings are ectomycorrhizal. So Boletus edulis, for example, matsutake, chanterelles, all ectomycorrhizal. And then some of the poisonous ones like the amanitas are ectomycorrhizal. But you also get things that you wouldn't think of as being ectomycorrhizal that look like they ought to be wood decay fungi. They're adhered to little scraps of wood or pine cones or needles or whatever. And they just form kind of a loose crust. And so a lot of these are ectomycorrhizal, and some of them are the most important ectomycorrhizal fungi in the forest. That is, they're the dominant ones on the roots. This symbiosis is highly convergent. On the fungal side, this paper from Hibbett and Matheny estimated a minimum of 8 independent origins of ectomycorrhizae just in the Basidiomycota, so just the mushroom forming things. This evolved 8 separate times. But just a year later, Teddersu's paper estimates that there's at least 66 independent origins of ectomycorrhizal symbiosis within the Basidiomycota and Ascomycota. And there are also some in other groups yet. The Mucoromycota has at least a few ectomycorrhizal fungi in it. So as complicated as this process seems to be, it was relatively easy to reinvent, and there are no good examples of evolutionary loss. That is, once a fungus lineage becomes ectomycorrhizal, there's no evidence it ever reverts to being saprophytic or parasitic. So it's really, this part of it is really a mystery, but there's pretty good data on it now. Ectomycorrhizal symbiosis are especially dominant in settings where nutrients are tied up in the litter. So when you get big areas like this with all this partially decomposed litter sitting there and very nutrient poor soil below it, that's a classic ectomycorrhizal soil forest setting. And the reason for that is that what these mycorrhizae do that the other major group of mycorrhizae don't do is that these can mobilize nutrients out of this litter that otherwise would not be mobilized. And I just realized this square should have been over here. It mobilizes the litter fungus. So, and they do this because they retain some gene content from their saprobic ancestors. That they have cellulases, they often have peroxidases, and they have lactases. So they can break down litter, and they're doing it not to get the carbon, because they're plugged into a big carbon source, which I'll get back to, but they're doing it because the most valuable commodity in this is the nitrogen. That in most of these settings, nitrogen is the limiting resource. It's tied up in this litter, and the fungi can access that for their host. So this shows some of that. These are little microcosms from the group up in Sheffield, England, Jonathan Leake and David Reed. And what you've got is a little birch seedling here, and it's growing in peat moss. And then here they've given it what they call a cafeteria experiment, where they put different types of litter out to see what the fungus will do when it reaches it. You can see that here the fungus is just gone crazy over this little patch of litter that it's given it, and likewise here. So by doing this sort of thing, they can show that they're mobilizing nutrients out of these patches of litter. Ectomycorrhizal fungi will do the same thing with rock, but they're picky on which rock they do it. So here we're showing pure quartz versus feldspar. It likes feldspar, it doesn't like pure quartz. Pure quartz doesn't have anything in it, but feldspar has potassium. And so they can mobilize some potassium out of the feldspar. And if we look at rocks in settings like this, they're often etched with fungal hyphae, or there will be boreholes in it. So there's evidence that they're actually directly penetrating rock and getting some of these nutrients. So how much does this cost? It turns out that one of the reasons that it's big woody trees is that it's an expensive symbiosis. So it's only plants that have a big resource pool that hire ectomycorrhizal fungi to find their nutrients for them. And the estimates are that 20 to 30% of net primary productivity from these trees goes directly to the mycorrhizal fungi. That's a big tax. And I always like to point out that it's a good argument for tax, because in spite of it costing this much, it allows these trees to grow places where nothing else can grow. So it's a good return on investment. Now this shows, this is back to the Leek and Reed group up in Sheffield. And again, we've got a birch seedling here. And what they've done is radio labeled it, and you're looking at autoradiography. And what you can see is that this is about 24 hours after they've radio labeled this. All the counts are already right at the mycelial front of this microcosm. So it's saying that the sugars that they fixed are just shunted straight out to the fungus, and the fungus is already reinvesting it out with exploring new territory and trying to find new nutrients. Okay, so that's the quick overview of ectomycorrhizal fungi, and it was we're going to use this in the next section, so hold on to it. And here's what you should remember, is that the relationship is obligate for most hosts and fungi. You don't get these things free living without them. The highest density of ectomycorrhizal forests are in the temperate zone, especially the boreal zone, but also southern hemisphere. It's only 2% of plants that are ectomycorrhizal, but it accounts for 60% of the trees in forests. There are over 20,000 fungi that are ectomycorrhizal, and the estimates are that this is convergently involved 66 times in the fungi. The main role of fungi is accessing organic forms of nutrients, especially nitrogen, and they charge the host a large carbon tax for this. So with that, I'll conclude this section on the dimensions of ectomycorrhizal fungal communities, and I'll open it up for questions.